Okay, so I have already recorded the video for Crackhead all to himself. However, I realized that Philosophical Tool brings up many of the same points and thought it prudent to do their videos not in tandem but one in the same. Plus, there were multiple issues I had with the script that I thought deserved a rewrite. This has caused a significant delay in putting out the video, but oh well. Cry me a river and build a bridge to get over it. This video will be split into 6 parts. The first is giving credit where credit is due. The second is addressing some strange comments or misunderstandings brought up primarily by Philosophical Tool. The third is addressing both Philosophical Tool and Crackhead's misconceptions about the science behind evolution and abiogenesis and more, and the alleged irreducible complexity that they think they've found. The fourth is the generalized claim they've both made about peer review being groupthink. The fifth is some questions they dodged, and the sixth is why they didn't debate me. Timestamps will be in the description down below. Credit where credit is due. Philosophical Tool brought up a point that I thought I should address because I got something wrong. While Michael Behe is indeed a proponent of intelligent design and irreducible complexity, he is, as it turns out, not a young Earth creationist. I said he was a biblical young Earth creationist. This is not the case. Here is the comment I'm referencing, where Philosophical Tool rightly points out that Behe isn't young Earth creationist. Just so everyone's on the same page, here are some quotes from a book Behe wrote called The Edge of Evolution. Evolution from a common ancestor via DNA changes is very well supported, and the bottom line is, common descent is true. It seems like his advocating for special creation lies only in specified areas where he believes God intervened to create a new system. I suppose this is slightly less terrible, but an unsupported belief nonetheless. Thanks for pointing that out. At one point in our conversation, I pointed out that I assumed he was young Earth creationist because he is both a proponent of intelligent design and a devout Catholic. Again, this was my mistake. However, look at his prior statement. You discredit anyone and or their arguments based on being a devout Christian. This did not happen. I didn't say he is wrong because he's a devout Catholic. I did not say his points aren't worth listening to because he's a devout Catholic. It was an observation about his religious beliefs which I believe influences scientific ones. There is no further inference to be made. When you have a hammer, every problem looks like a nail. Likewise, as Philosophical Tool assumes that I'm trying to be disingenuous, he looks for that wherever possible. Anyone with any sense reading the statement, he is a devout Catholic, will not infer anything extra. Another one. I pointed out that teachers refused to read a certain segment involved in the change to the Dover Area School District's curriculum when they tried to teach intelligent design. The part they were told to read and didn't goes as follows. The Pennsylvania Academic Standards require students to learn about Darwin's theory of evolution and eventually to take a standardized test of which evolution is a part. Because Darwin's theory is a theory, it is still being tested as new evidence is discovered. The theory is not a fact. Gaps in the theory exist for which there is no evidence. A theory is defined as a well-tested explanation that unifies a broad range of observations. Intelligent design is an explanation of the origin of life that differs from Darwin's view. The reference book of Pandas and People is available to students to see if they would like to explore this view in an effort to gain an understanding of what intelligent design actually involves. As is true with any theory, students are encouraged to keep an open mind. The school leaves the discussion of the origins of life to individual students and their families. As a standards-driven district, class instruction focuses upon preparing students to achieve proficiency on standards-based assessments. You can imagine how this is incredibly problematic to a biology teacher who has a functioning brain cell screaming, bro, this shit kinda whack though. For example, their assertion that gaps in the theory exist for which there is no evidence is completely untrue, along with, a theory is not a fact. Sorry, let's just teach everyone about humors and bloodletting because the germ theory of disease is a theory, or that Newtonian physics predicts everything because despite general relativity's predictions of the curvature of space-time or special relativity's predictions of the Lorentz transformations which has been proven, it's just a theory. That's absurd. The response from teachers, as previously mentioned, was a refusal to read it, as per Pennsylvania State Code 235.5a, Section B, Line 9, saying, in fulfillment to commitment to students, educators shall not knowingly or intentionally misrepresent subject matter or curriculum. Now that you're caught up on the lore. He said that because I pointed out this simple fact, which I thought was an interesting anecdote, that must mean I'm using it as an argument of some kind. Your little victory dancing about teachers not wanting to read it. No, not a victory dance. A fact. If you can't tell the difference between an interesting anecdote and a supposed victory dance, I think you should probably just take the L here and get that sorted out first. Again, if all you have is a hammer, every problem looks like a nail. Also, I got this sus as fuck comment from him. Furthermore, he accused me of using ad homs, short for argumentum ad hominem, which translates literally argument to the person, because you attack the person and not the argument. I did not. Just because you don't know what an ad hominem is doesn't mean I'm being fallacious. It means you're out of your depth. An ad hominem attack occurs when someone takes shots at someone's character as a way of dismantling their argument. For example, Billy is ugly, therefore he is wrong. I did not do this. I insulted Conman Behe multiple times in my video. This wasn't argumentum ad hominem because I wasn't using it as a logical argument. Saying you're an idiot isn't a logical argument because I'm stating a fact rather than using it to build a case. Rather, I discredited him and insulted him side by side. I wasn't using the insults to discredit him. They arose because I had discredited him already. He also accused me of strawmanning Conman Behe. This is not the case. Multiple times I used direct quotes from Behe himself. When I mentioned this to him, he ignored it. Another misconception he has about history is that the civilized world believed the earth was flat and that people ignored Copernicus. Firstly, not true. The civilized ancient Greeks knew that the earth was round. If you didn't know that, that's not my problem. More importantly, people ignored Copernicus in the 17th century because in 1616 his works were added to the Index Librorum Prohibitorum, the Catholic Church's list of banned books. Not epic, guys. Seriously. Let's start with this comment, which seems to imply he doesn't acknowledge general relativity or the existence of one-dimensional infinitely dense singularities. The short answer to this idea is that it's complicated. It seems like the reason Philosophical Tool opposes it is based on it doesn't make sense to me. Guess what, dipwad? The universe doesn't have to. The real reason why we don't know if it's true is that because despite the math predicting it perfectly, quantum field theory requires a discrete universe rather than a continuous one, which is to say a universe that can be divided into a smallest distance, which we know as the Planck length, as opposed to a universe that can be infinitely divided. If you have a universe that can be infinitely divided, well, you can have an infinitely small point and an infinite density. However, both relativity and quantum field theory are very well substantiated by every test available. This is a question that is simply unanswerable, at least for the time being, until we have quantum gravity, or in other words, a mechanism by which we can either simultaneously explain gravity as predicted by general relativity and quantum field theory, or at least offer an explanation as to how they are compatible. Unless Philosophical Tool knows something that the most brilliant minds in physics don't, he should stop talking. From here, we'll move on to a claim that was first made by Crackhead and further repeated by Philosophical Tool multiple times each. The claim was what Philosophical Tool called the protein paradox. The argument goes as follows. 1. You need protein to make DNA. 2. You need DNA to make proteins. 3. From 1 and 2, we can infer that they are two codependent parts of a system. 3.1. Therefore, taking away one will render the other useless. 3.2. Therefore, they must have arisen at the same time to allow them to function. 4. Darwinian evolution only allows one change to arise at a time. 5. From 3.2 and 4, we can infer that this system cannot have emerged from Darwinian evolution. It's a lot easier to attack when represented deductively, with premises and a conclusion. This is actually the argument for all areas of irreducible complexity, just with protein and DNA, so for whatever flavor of pseudo-scientific intelligent design you're pushing this time. For example, Behe's would be 1. You need certain proteins for the flagella to function. 2. You need other certain proteins for the flagella to function. 3. From 1 and 2, we can infer that these proteins serve as two codependent parts of the system. 3.1, therefore taking away one will render the other useless. 3.2, therefore they must have arisen at the same time to allow them to function. 4, Darwinian evolution only allows one change to arise at a time. And 5, from 3.2 and 4, we can infer that this system cannot have emerged from Darwinian evolution. Now let's jump back to this alleged protein paradox. I will show you two ways to attack this idea. First, we'll be attacking either premise 1 or 2 with some specific science. This is where I will be addressing exact claims they make. However, I will also show you in a generalized way how to dismantle any example of irreducible complexity by attacking a sneaky non sequitur later in the syllogism. Okay, let's attack the argument now. But we'll need some base understanding first. Before we start, a protein is just a large polypeptide that comes into play later. What is DNA? DNA, or deoxyribonucleic acid, is just a chain of nucleotides, but we'll get there. Let's start at the lowest level and build our way up. I understand that this might be a little complicated, but I'll do my best to explain it. Okay, so we're starting with the nucleobase. This is the part of the DNA that is unique. It's the part that changes, that stores information, and the part that is read from during transcription. We'll get there too, don't worry. Nucleobases in our DNA come in 4 forms: adenine, cytosine, guanine, and thymine. These are all unique molecules. Now we move up to the nucleoside. Nucleosides aren't that complicated, don't worry. It's just a nucleobase, one of those 4 chemicals, attached to a 5-carbon sugar. Boom. Simple, right? Okay, one more now, the nucleotide. This is like a nucleoside with an added one or more phosphate groups attached. Conversely, if this makes it easier, you can think of a nucleoside as just a nucleotide without those phosphate groups. You don't really need to know much about phosphate groups, it's just a term to describe a molecule with one phosphorus atom bonded with 4 oxygen atoms. The only thing that changes in a nucleotide is the nucleobase. All those other phosphate groups and sugars are the same in a strand, although depending on what nucleobase you get, you'll get either an A, C, G, or T nucleotide, depending on whether it has adenine, cytosine, guanine, or thymine as a nucleobase. Boom. Now we're so close to DNA. What most people know about DNA is that it's a double helix. Let's see how this happens, but first we'll look at just one helix. This is just a long chain of nucleotides, that's the phosphate, sugar, and nucleobase all neatly wrapped up, connected together by linking the phosphate sugar backbone. So how do we get from this single helix to a double helix? Well, the nucleobases actually link into base pairs. A links with T and C with G, and therefore conversely we have G linking with C and T linking with A. So what does DNA do? Well, first it is approached by RNA polymerase. This is an enzyme. It binds to the DNA and does some very complicated biochemistry where it reads the chain of nucleotides to get information. For example, it might read TCC, AG, TCA, etc., etc. And when all said and done, you have an RNA template. We call this mRNA, or messenger ribonucleic acid. This mRNA makes its way over to tRNA, or the transfer ribonucleic acid, which will decode the mRNA and convert it into a polypeptide, which, as we learned earlier, is a protein. This is the mechanism by which proteins are created in a cell, and without going too far into it, this mechanism is heavily regulated in eukaryotes by proteins themselves. Hence, we have this kind of chicken and egg problem. So how did this arise? Wait, let's take a breather and consolidate what we've learned so far. Nucleobases are either adenine, thymine, cytosine, or guanine. This is also the part of DNA that is read by RNA polymerase to generate the proper mRNA strand. One level up, we have nucleosides. This is a nucleobase attached to a 5-carbon sugar. Above that, we have nucleotides. This is a nucleobase plus one or more phosphate groups attached. The nucleobase will determine whether it's an A, T, C, or G nucleotide. Moving on, again, how did this actually arise if they require each other to function? Well, firstly, we should do well to note that DNA probably wasn't the first nucleotide chain to arise. Rather, RNA makes for a much better candidate, hence the popularity of both the RNA world hypothesis and the more recent RNA peptide coevolution hypothesis. The reason for RNA being such a good contender is that RNA can store information like DNA and act as a catalyst or enzyme like proteins. It's almost like the best of both worlds, but there are some drawbacks which we won't cover here. Let's just cover RNA for a second here. RNA is extremely similar to DNA, and in fact shares 3 of the same nucleobases, with thymine sold for uracil, giving us A, U, G, and C nucleotides, but A nucleotides can still pair with U nucleotides, which makes double-strand RNA technically possible, although highly rare in nature. Furthermore, double-strand RNA, or dsRNA for short, shares many properties with DNA that single-strand RNA, or ssRNA, doesn't. For example, it is much more resilient and can survive in conditions where ssRNA will completely break down in minutes. The other notable difference is that the sugar on RNA is ribose, whereas on DNA it's deoxyribose, hence the ribonucleic and deoxyribonucleic acids. Okay, moving on. Firstly, we know RNA nucleotides may have existed at the beginning of the Earth, as demonstrated by a paper linked below entitled Spontaneous Formation and Base Pairing of Plausible Prebiotic Nucleotides in Water. Very nice. A quote from the end sums it up nicely if you're not bothered to read the whole paper. The data presented here demonstrates the efficient single-step synthesis of complementary nucleosides and nucleotides, starting with the plausible protonucleobases melamine and BA and ribose or R5P. On our hypothetical prebiotic Earth, we now have RNA chains floating around. Moving on, we must figure out how cells emerged. I find this area really fascinating. This comes from the work of Jack Szostak, a Nobel laureate, and someone who has summarized his work in a YouTube video linked below. However, there is a longer 3-part series which he presented himself if you want more specifics. So another thing we know is the existence of fatty acid lipids in a prebiotic Earth. But hold on, let's define that too. A lipid is just an organic compound that doesn't dissolve in water. You can imagine how this would serve as a nice protocell for self-replicating nucleotide polymers, and in fact it does. As these vesicles encounter other free-roaming fatty acids, they absorb them. Growth is driven purely by thermodynamics. Furthermore, these fatty acid vesicles divide when they get big enough due to mechanical forces like rocks and currents. This is essentially a self-replicating cell without the genetic information. So far, we've got both the capacity to grow and divide, along with the capacity to spontaneously form nucleotide chains and even base pairs. How does this tie together? Well, first, let's define a polymer. A polymer is just a chain of monomers, which is basically just a form of organic compound that can form a chain. So how does this all tie in? Well, there's a little more information that I've carefully withheld until now. You see, those vesicles are what's called semi-permeable membranes, which allow small molecules in but not larger ones. So nucleotide monomers move into the membrane, polymerize into polymers, and bam, trapped inside of this protocell. Here's where it gets cooler. These polymers increase the osmotic pressure inside the vesicle, making it stretch to be larger. A vesicle with more polymer inside will actually steal lipids from a vesicle with less polymer, making it even larger. Now we have competition and natural selection, with these lipid vesicles eating each other, and when they divide, the nucleotide polymers inside will go in both new vesicles, essentially a basic form of genetic reproduction. You now have evolution by natural selection, with the most self-replicating polymers being selected for. Current theories posit that an RNA enzyme known as a ribozyme that was capable of copying RNA molecules existed at this early stage. From here, it's easy to see how protein synthesis via the ribosome, which is a kind of RNA enzyme, evolved out of this system. We have now established the credibility of an RNA world beyond reasonable doubt. This is an important step because now we can establish how we move from RNA to DNA. In an article entitled Origin of Life, Transitioning to DNA Genomes in an RNA World, the author rightly points out that now, in eLife, Joyce and Biswajit Samanta at the Salk Institute demonstrate that this ribozyme is also a reverse transcriptase. Feeding DNA nucleotides to this ribozyme enabled it to copy short segments of RNA templates into complementary DNA. This suggests that if an RNA world contained DNA nucleotides, DNA genomes could have assembled and then presumably replicated by ribozymes. So that begs the question, could DNA nucleotides have been present in an RNA world? The result is an astounding yes. In the article referenced previously, he links multiple peer-reviewed papers, only one of which I will be mentioning here, although there are plenty more. This paper is entitled Conversion of Biosynthetic Precursors of RNA to Those of DNA by Photoredox Chemistry, linked down below, and contains a nice summary in the Results and Discussion section. We will just be looking at that for now, where this relevant sentence comes up. It was found that there was 42% residual ribose and that 2-deoxyribose had been produced in 52% yield, leaving only 6% of the starting material unaccounted for, presumably as minor unidentified products. A 52% yield! So, to recap, we have established a mechanism by which we can have an RNA world which includes competition and therefore natural selection. Then we moved on to establishing how proteins would arise out of this RNA world. Moving on, we showed how not only if DNA nucleotides were present, how this would lead to the production of DNA gene sequences, but also in fact that they were present. Okay, argument dismantled. But he had another that he would not shut up about. The development of motor proteins and their interaction with the cytoskeleton. Again, let's frame this as a syllogism. 1. You need motor proteins for the cytoskeleton to serve a purpose. 2. You need the cytoskeleton for motor proteins to serve a purpose. 3. From 1 and 2 we can infer that they are two codependent parts of a system, blah blah blah, you've seen it all before. This is a pretty obvious problem and requires way less biochemistry, but first let's define our terms like we did last time. The cytoskeleton is a structure in cells made of filaments or fibers present in cytoplasm. It serves a multitude of functions, however, by far the most important is serving as a track for motor proteins. But hold on, what's a motor protein? Motor proteins actually come in 3 flavors: kinesins, dyneins, and myosins. All 3 of these motor proteins have the ability to carry cargo in a cell and walk along the cytoskeleton as a sort of train track. Kinesins and dyneins run on something called microtubules, and myosins run on something called actin filaments. This may seem irreducibly complex on the surface, but I assure you it's not. It's actually very simple to debunk. The problem here is that the cytoskeleton serves functions outside of motor protein tracks. For example, the cytoskeleton is responsible for maintaining the shape and structure of a cell. 1. You need thing X for thing Y to function. 2. You need thing Y for thing X to function. 3. From 1 and 2, we can infer that there are two codependent parts of a system. 3.1, therefore taking away one will render the other useless. 3.2, therefore they must have arisen at the same time to allow them to function. 4, Darwinian evolution only allows one change to arise at a time. 5, from 3.2 and 4, we can infer that this system cannot have emerged from Darwinian evolution. The problem here comes from 3.2. The underlying assumption ignores the fact that the following sequence may have occurred. Thing Z does function 1. Thing X evolves to help with function 1. Thing Y evolves to combine with thing X to do function 2. Function 1 is rendered useless. Because function 1 is useless, thing Z devolves. Now you have thing X and thing Y doing function 2 with a seemingly irreducibly complex system. In response to Michael Behe, a man named John McDonald developed a mechanism by which a mousetrap could be reducibly complex. A mousetrap, of course, being Behe's favorite analogy for an irreducibly complex system. The link to that down below, but a quote worth mentioning is found at the top. The mousetrap illustrates one of the fundamental flaws in the intelligent design argument. The fact that one person can't imagine something doesn't mean it is impossible. It may just mean that the person has a limited imagination. Well said, my friend. Well said. Groupthink. The term groupthink, as used today, was coined by Irving Janis of Yale University in 1972. Groupthink refers to when a group of highly intelligent people ignore proposed issues from other people. For example, if they all have the same background, it may insulate the group from outside thoughts. A good example of groupthink was the 1986 Challenger disaster. The engines that made the solid rocket boosters warned NASA scientists that the O-rings that held the boosters together were frozen due to cold weather the night before, and that to launch is a terrible idea. The NASA scientists, in a sheer display of arrogance, launched anyway. The rocket broke apart and exploded 73 seconds into the flight, ultimately killing all 7 members on board. Both Crackhead and Philosophical Tool have made the claim that peer-reviewed papers are groupthink. This is not the case. If you think this, it is because of your lack of understanding of peer review and the scientific method. Groupthink occurs when the group stops criticizing each other and instead just goes on with what each other says because it fits their ideas. This is the opposite of peer review and in fact takes place in intelligent design communities, like when Christians go along with whatever conman Behe says just because it fits their ideas. Peer review is the process by which academics in a field will review a paper you write prior to it being published to ensure that the paper is accurate and reliable, and the experiment conducted is both clear and repeatable. The entire point of peer review is to weed out groupthink. I think we can all agree that if a large number of papers are rejected via peer review, it cannot be groupthink, because groupthink would only occur when ideas that propagate the ideology are accepted regardless of intellectual honesty. According to authorservices.wiley.com, 40% of papers are rejected after the peer review phase, which clearly indicates that papers aren't being accepted on the basis of fitting the narrative, but rather clear and consistent science. So there you have it. Calling peer-reviewed papers groupthink is just projection from intelligent design proponents who don't want to admit that they're wrong and would rather just smear science as a whole because it demonstrably shows how their beliefs are just as archaic and outdated as flat earthers. Dodging questions. Let's start with Philosophical Tool talking about the lack of precursors to the Cambrian explosion. I asked him to clarify this more than once. I still do not understand what he means by this due to his refusal to explain himself. I told him that if he did not clarify, my assumption would be he means causes of the Cambrian explosion. This is what I will assume he meant, although he is a master question dodger, so I can't be sure. If you must know, there are multiple factors which caused the Cambrian explosion. A few are an increase in oxygen levels, the formation of the ozone layer as to protect land animals from lethal UV rays, an increase in calcium concentration of the Cambrian seawater, the increase in predation intensity, thus creating a sort of arms race between predators and prey. There are many more, but these are the main ones. Reused proteins. In my debunk video on conman Behe, I rightfully pointed out that he ignores the simple fact that proteins in the flagella are actually reused from other systems, making them not irreducibly complex. In response, Philosophical Tool says, "Lol, you argument about reusing protein has already been tried and has been refuted successfully." I asked him then to cite a source as I cited mine. He did not. I did not get any response. He did not tell me where he got his information, which proteins were or weren't reused. He then just went on his little rant about my supposed victory dance. So why didn't they debate me? By now, I think this should be clear. They don't want to debate me because they're out of their depth, because they don't know what they're talking about, because I do, because the science is on my side, because at the end of the day, all that matters to them is holding on to their precious archaic beliefs instead of growing up. Let's look at what they say. Crackhead constantly insults me, saying it would be like child abuse or picking on the mentally disabled. Philosophical Tool also said multiple times that I didn't want to debate him, presumably because it would be oh so crushing. I mean, come on. These are like drunk guys at a pub who say, "Oh no bro, I'm so strong, you don't even want to fight me, I'll actually fuck you up." "No bro, I'm not gonna fight you for your own safety." Everyone knows what's going on. You won't debate me because you're cowards, because you're wrong. You just want to save face and make it act like it's for my own good. I have never seen a bigger pair of clowns in my life. Okay, here we are. The end. There's not much more to say. If anyone would like to debate me, my email is always open along with the comments section. A little message to Philosophical Tool or Crackhead or anyone who wants to leave a comment. If you want to comment on this video, it must take one of 5 forms. 1. It can be a constructive comment on the state of the video, i.e. the editing, the audio, whatever. 2. It can be a constructive or critical comment about the science I have brought up. 3. It can be a question about me, about the science, about anything, so long as it is asked in good faith. 4. It can be an apology or a debate challenge. 5. It can be another way of interacting with the video, perhaps a recommendation, as long as it stays positive. Here's what it can't be. It cannot be unnecessarily vitriolic. Any comment by anyone from here on out will be deleted without warning if it contains unnecessary insults or otherwise demeaning messages to me, whoever I'm responding to at the time, or to anyone else in the comment section. This is a hard rule. No one will be getting away with this. It also can't be another argument for irreducible complexity because I've already debunked it. I'm not doing it again. I'm not wasting my time. Complaints about censorship may or may not be deleted depending on the form they take. If they're respectful, perhaps we can have a dialogue about that. If you're going to compare me to Stalin or the Soviet Union, I'm just going to delete your comment. Otherwise, happy hunting, have a great day, and hopefully I'll see you next time where we make a video responding to Matt Powell.